Hello, and a warm welcome to everyone. Thank you for joining us. For those of you not familiar with us, Inscopix is a neuroscience tech company in the heart of Silicon Valley, and we provide complete miniature microscope solutions to scientists that want to map brain circuits in freely behaving animals. We are thrilled to bring back the Insights webinar series to the neuroscience community. We opened this webinar series led by world-class neuroscientists at the start of the pandemic in 2020 with the intent of providing a sense of connection and community across the world to those who may feel distanced during these challenging times. We hope everyone continues to stay healthy and safe as we navigate through 2022. Before we begin, I just want to mention that attendees can submit questions at any time using the Q&A box below. We will be collecting questions throughout the webinar and will answer as many as possible at the end of the presentation. We do have a hard stop at the end of the hour, so if there are questions that we don't get to, please know that it is only due to lack of time. With that, it is my absolute honor and privilege to welcome our first Insights webinar speakers of 2022, the illustrious Dr. Richard Morris and his research colleagues, Dr. Francesco Gobbo and Rufus Mitchell-Heggs. Richard Morris is a professor of neuroscience at the University of Edinburgh. In addition to being known as the developer of the Morris Water Maze, one of the most widely used cognitive assays in behavioral neuroscience, Richard is known for his work on the role of N-methyl D receptor activity in spatial memory. He has recently embarked on new projects in social neuroscience to complement his other research. With Tim Bliss and Graham Collingridge, he won the Brain Prize in 2016 and is an elected fellow of the Royal Society in the UK, the Norwegian Academy of Science and Letters, and the National Academy of Science in the US. We are thrilled to have him join us today. His colleague, Dr. Francesco Gobbo, will also share some exciting research from the Morris Lab. Francesco graduated from the University of Pisa and the Scuola Normale Superiore Excellence School and received his PhD in neuroscience in 2018. Francesco's expertise lies in molecular, cellular, and behavioral neuroscience. His current interest is in the representations of episodic-like memories in the hippocampus system and how this information is used to guide behaviors. Since joining the Morris Lab as a postdoctoral researcher, he has taken a specialized interest in using Inscopix miniscopes in rat hippocampal CA1. We will see some of his research highlighted here today. And last but not least, we have Rufus Mitchell-Heggs. Rufus has a master's in neurotechnology and is currently working towards a PhD in neuroscience from the Center of Discovery Brain Sciences at the University of Edinburgh in collaboration with the Center for Neurotechnology at the Imperial College London. His specialty lies in neuroanalysis, the application and adaptation of statistical and machine learning methods to process and analyze the ever-growing quantity of neural population and behavioral data. Today, you will see some of his research highlighted here. Gentlemen, I want to thank you all for joining us for this webinar. So without further ado, I'm going to turn this over to you, Dr. Morris. I will let you navigate the screen share from here.
So let's see. So I hope people can see that. Let me start by saying what a pleasure it is for all three of us to have a chance to contribute to this wonderful series, which I think really has done a lot to keep people connected during these difficult times across the world. So my congratulations to Inscopix. It's a pleasure to be here. We plan to talk about some of the research we've done and some of the problems we've faced, because we've been very much on a learning curve using the endoscope and using the software. And we may well be still a bit at the bottom of that learning curve. We'll see. But I want to say that it's a pleasure to be able to present this work. But if there are additional questions that people would like to pose, please don't hesitate to do so. So our scientific aims relate to two projects that we want to discuss. First of all, discussing how we've gone about using in vivo calcium imaging in both the prelimbic zone of the prefrontal cortex and in hippocampus. The fact that we use rats rather than mice as experimental subjects. And in the time we've got, we want to tell you a little bit about a social neuroscience project that I've become involved in, whose overarching aim is to look at the neural basis of social deficits in autism spectrum disorders such as Fragile X. And then I will pass over to Francesco and Rufus, who will talk about a spatial memory project whose overarching aim is to identify the role of place cells and other hippocampal cell types in navigation from one place to another. Now, with respect to social neuroscience, we're in, I think, a very exciting point where there's a real opportunity of making serious advances, particularly with monogenetic causes of autism spectrum disorder, of which Fragile X is one example, SYNGAP another. And we have in Edinburgh one of the world leaders in neurodevelopmental disorders, namely Adrian Bird, who's been working on Rett syndrome for many years. And Adrian, together with my colleague Peter Kind, has been instrumental in creating a Simons Institute for the Developing Brain, in which both human clinical studies and animal work is going on. And of course, one of these has to do with FMRP and Fragile X. Another, as I mentioned, is to do with SYNGAP, and there are various animal models that we're pursuing. Now, with respect to the second part of the talk, the place cells, I think we all know what place cells are if you have any interest in the hippocampus as boundary vector representations of location as shown in the cartoon A. But one of the puzzles about place cells is the puzzle of how these cells might be used in navigation. Because if a place cell is defined as a cell that fires if and only if an animal is in a particular location, say place A, then how does the animal in place A access information about place B, which will have a different set of place cells, or place C? The question of how that is done has been of great interest to lots of people over the years. Some wonderful work done by Bruno Pousset in France, computational models by Daniel Bush and Neil Burgess at University College London. And just 2 weeks ago, Hugo Spears and colleagues published a wonderful review in Neuron about the problem of goal cells. So let me just turn to the social neuroscience project. We have been using the tube test of social dominance, on which there's been lots of work done on mice, but it works equally well in rats. It consists of two holding boxes shown in the cartoon that you can see, with a perspex or plexiglass tube connecting these through which the animals can move. So you have an animal coming from the left and an animal coming from the right. And what we have done in a number of different experiments, of which this is just one, is trained animals just purely in a behavioral task to ask the question of kind of who is dominant. And in this particular study, we were interested in Fragile X animals versus wild type controls. And we measured the hierarchies within individual cages, and we also compared animals in one cage with animals in another cage. Now, a key point is that we had 7 cages of 4 animals per cage, of which half the animals were wild type and half were knockout. And that enabled us to work out who was dominant. So you can have rank 1, 2, 3, or 4, as shown on the y-axis, across a set of 10 sessions. And this was pretty typical data that we got, namely that the wild type animals won over the knockout animals. The effect was highly significant and was measured just in terms of the number of wins using a chi-squared test or the average rank using analysis of variance. Now, we obtained similar results in SYNGAP, but I don't want to actually dwell on the individual data, but to raise the question of where we went from there. And here I was greatly inspired by the work of Hailan Hu, who many of you may know, in Zhejiang in China, formerly a member of the Shanghai Institute for Neurosciences, who published a series of papers during the 2000 to 2017, providing strong evidence that the prelimbic zone of the medial prefrontal cortex played a causal role in mediating relative dominance amongst animals. Her studies were very thorough, and even the behavioral ones were able to investigate whether the relationship between, say, animal A, B, and C was transitive or non-transitive. Then they went on to do very elegant slice physiology and more recently optogenetics. So inspired by those set of results, we decided to focus on the prelimbic region. And as many of you attending this will be fully aware and even using them yourself, we decided to use the NVISTA endoscope, which you can see in cartoon A, directed at the prelimbic zone of the rat brain. We're going to express GCAM6F in that zone and target the grin lens to that particular position. And you can see a camera view shown in camera C. In addition, we did various amounts of histochemistry to look not only at the expression of GCAM6, but also various neuronal markers, NUN, DAPI, GAD67, and of course the co-localization of these. Now, since we've got a social situation with two animals interacting, we face an interesting problem. And that is that if each of these animals is, as it were, wearing a helmet in their base plates, then there's every chance that animal A will interact with animal B and may even damage the camera on the other animal. And clearly we didn't want that to happen. So what we decided to do was to create a helmet to protect the camera, and we call it the eye helmet. And it is placed around the camera and in a way that is also around the base plate that normally holds the endoscope. These helmets are built using 3D printing in the lab. They're all made in-house. And what you see top left is the surround, the sort of base plate surround, which is placed on the animal and kept there permanently. It has a little cap shown in middle top, which protects the implant for protecting the endoscope itself, so that when the animals are in their home cage, what it looks like on the animal's head is like the top middle picture. Now, the helmet itself is a separate unit which can be attached when we take this protective helmet off, and we can print these in various colors like red and green or what have you. In addition, there's a cable catcher, which is shown in black, so that if there's any tugging of the cable, the force of that tug will be transmitted to the helmet and not to the camera. Now, finally, the inside dimensions of this helmet are 1 to 1.5 millimeters wider than the widest and longest dimensions of the camera. So that if one animal were to knock the second animal, it would be knocking the helmet, but would not knock the camera. And this device, this simple little bit of plastic, has been published, 3D printing solution for safe endoscopic recording, in the Journal of Neuroscience Methods, and some of you may be aware of that paper. And an important result that we found was that if we looked at how stable the recordings we got without the helmet and compared those to with the helmet, you can see that we were able to massively reduce the number of motion artifacts. Now, this is clearly very useful in a social situation, but I hazard it may be valuable in other situations as well. So we throw that out to you. Now, here's the situation. Here's two animals in the tube about to interact, one with a green helmet, one with a red one. And Deep Lab Cut is tracking the head cap of the two animals, so we've got an idea of their sort of location. And these animals decide, you know, kind of what's about to happen here. And you can see simultaneously the calcium recording that we're getting. Now, we only have one camera in this project, so there's actually a dummy camera in one animal, but we swap these around from animal to get stable data. Now, in a set of 12 rats that we've looked at so far, 6 wild type and 6 Fragile X, we've secured 900 stable longitudinally regions of interest, i.e. cells, averaging about 75 per rat. Now, in panel A here is shown that longitudinal registration. You can see a couple of bright regions of interest for session 1 in panel A, showing up again in session 3, one of them showing up in session 5, another in session 7. In panel B, the neural activity, delta F over F, that we get from individual regions of interest. And then plotted in panel C is the mean delta F over F across a large number of cells. It may be an average of 75, sometimes more, sometimes less. And you can see that mean value normalized to the highest value of delta F over F we get. So the black line going up and down is just the mean activity. Now, what are the colors here? The colors refer to the specific behavior that the animals are executing. So they may be moving forward in gray, they may be pushing in a sort of pinky red color, resisting, retreat in a light green, or they may be just still. And it turns out stillness is actually a very interesting pattern of behavior, which we're trying to study more, or they may just withdraw once the contest is over. Now, when we look at these average data, we can ask the question of whether the mean neural activity, delta F over F, is any different in winners or in losers, which addresses the issue of dominance. And what we find is, we think, a potentially interesting result, which is that if you have one animal who's strongly dominant, it will be showing a mean value of about 0.55, which stays stable over a series of trials. But the clear loser will be much lower at 0.35, dropping down to about 0.25. So there's a very clear difference in mean total or mean activity between the strong winners and the strong losers. But for the moderate winners and losers, actually the trend is, if anything, that the loser shows greater activity, but they don't differ significantly. So now that's just the delta F over F measure. When we then turn to looking for events, what we will be doing is going from delta F over F, shown in black at the top, to taking out the noise and then identifying the events using CNMFE. And that enables us to find out a precise moment in time when there may be one cell or burst of firing from various cells. And this data was obtained by our colleague Nuria Garcia. So when we derive such events, we then want to ask the question, not just of delta F over F, but of the event record. Is there any relationship between the cell firing and behavior? And one of the first findings to share with you is that we do observe what we call unique behavior cells. And this is an example for the case of retreat. So the animal retreats during the sort of light green periods and not at other times. You can see we've got 6 cells here which show events. And although cell 2 does show some firing outside the light green pattern, the basic picture that comes across from this is of a striking relationship between the activity of retreat and the activity of these cells. Now, it's all very well to show a good exemplar like this, but can we get any mathematical precision on this? Well, that was a key point. And when we submitted a paper based on some of this work, the referees, I think, quite rightly came back to say, could we actually provide some mathematical justification for this relationship? So what we did was to move to calculating what's called the mutual information between cell activity and behavior activity. So just to take you through the gist of this, because I don't have a lot of time, we've got a behavior, call it B1, B0, which would be a different behavior, then back to B1 again, and so on. And we can ask the question of, do the events happen only during B1 and not at other times? And in the top line in black, you can see there's a pretty close relationship. But then we shuffle the data 2,000 times to create a frequency mutual information graph, which is shown at the right, and ask the question of whether the value of mutual information that we get varies as a function of the shuffling position. And what we find is if we get to a mutual information or K value that's above, say, the 95 or the 99th percentile, then we feel we're justified in claiming that there is a mathematical relationship between those. And that's how we're trying to do this. So, is retreat a unique example, or can we find other examples? Well, shown here on the slide is a push cell, cell 121 in animal 518, a resist cell, cell 25 in animal 686, another retreat cell with different sort of patterns, cell 54, and a withdrawal cell. So I think we're moving to a position where we'll be able to give many examples of this and provide some mathematical justification for claiming uniqueness, and also compute how many of these sorts of cells there are and how many are much more non-specific in their Farring pattern. Now, this is where we're looking at the behavior of the animal from whom we are recording. However, bear in mind, this is a social situation. We've got two animals, one confronting the other, and this raises the question of whether the activity in the animal from which we're recording is correlated only with his own behavior, or might be correlated with the behavior of the other animal. So we therefore looked at our cell firing in relation to the behavior of the other rat. And here I present to you firing in animal 522, when the animal with whom he was confronting was a different animal, and that animal was pushing. And then the other animal was resisting. The other animal was retreating. The other animal was withdrawing, and so on. So we find this a very interesting correlation. In some sense, it's sort of obvious. In a social situation, the brain would respond to the behavior of the other animal. But of course, this is quite different from the usual way in which so much neurophysiological recording is done, where the emphasis is on the animal from which the recordings are being taken. Now, we should make the point in passing that similar observations have been reported by Kingsbury et al. In Cell in 2019. They use the delta F over F measure rather than discrete events, which we've got after CNOFE. But I'm not sure whether that matters too much for their claim that the correlations to the other animal can be seen. But we do have a wider range of behaviors and a wider range of examples. So that's really what I wanted to present to you. And I think now we should, without further ado, move on to Francesco and Rufus, who have a very different project.
Thank you, Richard. Let me just share my own screen. Right. So the project that I've been mainly involved on, as Richard was saying, is a work on spatial memory and episodic memory in particular. So this project was started by me and another very gifted and dear postdoc in our lab, who is now at Edge Hill University, and Dorothy Sem, and Rufus here that has worked with us on basically the entirety of the project. Now, for those of you who are not familiar with place cells, just a few words. So in general, the hippocampus is involved in spatial representation and memories, and in particular, episodic-like memories or episodic memories in humans, which basically are the type of memories that contain information about what, where, and when. So basically, complete memory with spatial and temporal contributions. So place cells are neurons that fire preferentially where the individual is in a particular point in space, and although they have been originally being recorded in neurons, they have been found also in humans. And so their contribution in navigation has been demonstrated with multiple experiments, and for example, disrupting the place field of place cells by electrophysiological means or others impairs the performance of animals performing in a task. But what's interesting for us is, and what Richard was mentioning in his introduction, is how is the play cell activation involved in representing the planning of future actions by the animal. So there has been evidence for some correlation between the sequential activation of play cells and the path the animal are about to undergo and undertake in the future. And so but however, overall, the role of the hippocampal activity in planning the future action of the animal is still actively debated. So that's where we decided to start our project. And so our questions are basically, we were gathering answering this question to use the miniature endoscope by the Inscopix, because that would allow us to monitor the activity of cells in the hippocampus over multiple days, and while the animals were free to perform in without constriction due, for example, to head restriction or other reasons. So our first question is then, how is the spatial information is represented in the hippocampus of rats when this is recorded with calcium imaging, for example, as opposed to electrophysiology, which is, if you want, the gold standard in recording the neuronal activity in the hippocampus of rats. And then moving to our main question, which is basically how the spatial information that the hippocampus contains is used to retrieve memories and to guide the decisions about choices that animals make. And last, how the spatial information can be modulated by other tasks, like, for example, if animals can perform the same task using different strategies, if the type of strategies that is chosen or learned by the animal can influence how space is actually represented in the hippocampus. So the behavioral model that we're using is the everyday arena. So the everyday arena task is in practice a two-dimensional reward task, and it is composed of a series of sessions where each day or each session in general, the animal learn to retrieve a food reward, so a positive reward from a different location, which is basically a sandwell, which is located in the floor of the two-dimensional arena. So as we're saying, the episodic component is mainly involved by the fact that the position changes from day to day. So in our task, each session is composed by three parts. So the first one is the pre-training, where the animals explores the arena in absence of rewards, and that allows us to record the play cells in each particular session without the presence of rewards. After the pre-training, the animal enters the sample phase, where basically the animal enters the arena from one of four possible start boxes and has to find which one among the possible locations is the rewarded one in that particular session. So in practice, the animal exits the start box, entering then the arena, has to find the correct location, retrieves food pellets, and then he has to come back to the original start box to eat the pellets, and then he has the chance to come back again and get a second and then a third pellet. After 45 to 60 minutes, that's what we call the choice trial. So the animal is then presented to indistinguishable sandwells in the floor, and because they're covered in sand. And starting from the start box, the animal has to remember which one is the correct location in that particular day. So that's how we test the memory. So over time, the animal learned how to perform the task, and you can see like a pretty nice learning curve over time, over the number of sessions. And while initially the performance, so the number of errors that the animal commits before reaching the correct location in the choice trial is basically a chance. Over time, saying from session 8 to 10, basically, the animal are proficient in correctly remembering which one is the correct location, and that is paralleled by a decrease in the latency in reaching the correct location. So that's basically the time that the animal takes from going out of the start box and reach the correct location. of course, we check for a number of confounding factors. For example, we check that the animals are not using olfactory cues in probe trials, which are essentially identical to choice trials, but in absence of reward present in the choice trial, and we just calculate how much time the animal spends digging in the correct location without the confounding factor of olfactory cues. Now, we then use this paradigm and we try to apply the calcium imaging using the Inscopix miniscopes to the rat CA1. And when we first started, we only had the information about the experiments performed in mice, essentially. And however, when we moved to rats and we first tried to apply that in rats, we found that the rat CA1 poses particular challenges to imaging, which are not necessarily the case in mice. And the most evident one, if you want, is the presence of a very thick layer of fibers that basically involves the outside of the hippocampus, and it's basically overlying CA1. So in order to do that, after aspirating a portion of the cortical tissue above, we are then aspirating a portion of the fibers on the location where we are implanting the lens, and of course taking care not to damaging the outside stratumorians of CA1. And that's when then we allocate our green lens, which will be then used to image in CA1. And here, for example, you can see three animals that show the amount of tissue that we aspirate on top of the hippocampus. So you can see that the stratumorians is essentially intact, while we have aspirated both the cortical tissue above it, but also the fibers on top of the location where we are going to implant the lens. And of course, we checked that the expression was restricted to excitatory pyramidal neurons in CA1, of course, of the calcium reporter, which is in our case GCAM6F. And so typically we're that allows us to record from a number of cells that depends on the animal, but it's between 100 and more than 300 per session on a typical 20 minutes recording. And that's typical fields of view that you get from using the procedures that we developed for the rat hippocampus. After the recording, we use the Inscopix program to process our raw video. And so from the raw video, we first correct for the motion, we subtract the background. And after reaching the delta F over F stage, we then detect cells using CNMP algorithm and then the events DOASIS algorithm. And so as after the correct pre-processing of all this data, we're able to typically record and align longitudinally cells across a very long period of time. So in our experience, it can be, we can find the same field of view over a period of a month or even longer, typically. So you can see, for example, on the right, on the left, that's one example of our animals that we included in the analysis that we are presenting, and you can see the same field of view over 30 days. So from session 1 to session 20, which is about 30 days later. And this is important because we are comparing the performance and the representation of place cells, but also of cells which might be involved in goal location using different session where the location of the reward changes. And then I'll just leave it to Rufus to present the subsequent analysis from the pre-processing we did.
Great, thanks, Francesco. So fantastic. Francesco has just sort of spoken through exactly how we build up the story from the experimental side and also the calcium imaging side. And now we're going to get into the questions which he outlined at the start. So how does this hippocampal CA1 neurons encode decision-making? So essentially we have our rat, which is located in its start box, and it has to decide which rewarded well it's going to head to. Two of them don't have the reward, one of them does. And at some point, it has to decide how it's going to do this. It has to recall this information. So the way that we aim to do this is to look, first of all, and see how the spatial information is represented in the hippocampus of rats when recorded with calcium imaging, and then to look into how spatial information is used to retrieve memories and guide that decision making. So the way in which we did this was, first of all, to extract the location of the animal in time. So we use deep lab cuts as before that can recognize the animal and where it is in time. And we can sort of start building up this picture of where the animal has been in different times. We then look at the neurons and we can extract the neural traces, and we align these two, this behavioral activity to the neural activity. And what we can start doing is, for individual neurons, start seeing where that neuron is firing in the arena, what is the sort of place that it is focusing in on. And basically we use something which is a place cell classification pipeline, similar to what Richard mentioned before, but just a way for us to start correlating a cell's activity to a specific location in the arena. So we end up doing this for all of our cells, and each one of these is a different cell, and each box is essentially the arena. So essentially where you're seeing higher activity or closer to 1, that's where that cell is sort of having its peak activity, and where you're seeing sort of darkness, that's when the cell is not active. So you can see that, largely speaking, a large proportion of our cells are starting to encode the spatial location, the animal's spatial location in the arena. The next thing we wanted to do was look at this from a more population perspective. And here what we're looking at is the sample trial. So this is where the animal is leaving the start box and learning for the first time where the rewarded location is. And then it'll pick up a reward and then go back to the start box. So similarly as before, what we're able to do is track the animal's position going in and out of the start box, and we can align that with the neural activity of individual neurons and the population as well. And so what we do is essentially, for each trial, so if every time the animal exits the start box and then re-enters, we are able to look at the neural activity within that trial. So that being what neuron 1 is doing at time point 0, what neuron 2 is doing, neuron 3. And we can start building a picture of what the whole neural population is doing during that trial. And what we do is we compare the neural population activity within that trial and across trials using a measure called the cosine distance. So we take the distance between neural population activity in different time points, and we can build this adjacency matrix that we've got here. So here on each of the axes, we're comparing different trials with themselves and other trials. And down the center, you can see there's this big dark strip, and this corresponds to a very low cosine distance, which basically means that the neural activity is very similar, if not the same. So this central one, we're basically looking at trial 1 compared with trial 1. So clearly the neural cosine distance is going to be 0 because the population activity is the same. Whereas if we look at 1 and 2, we start seeing that actually there is similar neural activity as well. We have this dark strip for 1 and 2, and also 1 and 3, 1 and 4, 1 and 5. And what this tells us is that the neural activity that's occurring in the first trial of the animal leaving the start box and coming back in on itself is repeated across subsequent trials. So there's another way which we can look at this. It's maybe a little bit more clear. It's using a technique called neural manifold learning, and in particular, we're using something called multi-dimensional scaling. In this instance, we can separate out each of the trials and see what the individual manifolds look like. Each point on this manifold represents a point in time, and the distance between the points represents the difference in neural population activity. So the closer the points are, the more similar the neural activity, and the further they are, the more different they are from one another. What's quite interesting is that you can see for each trial, we have this loop-like activity. First of all, starting with the start box activity here at the bottom, and then going as the animal traverses through the arena and over to the reward well activity, you can see that it is conserved across the different trials. So we have the same neural population activity going on across trials. And this implies that there's similar neurons, like place cells, that are firing across trials. The next question we wanted to ask is, when is the decision-making process taking place? So we have our animal in the start box here at the beginning, and we were able to train a naive Bayes decoder using neural activity solely in the start box as to and predict where the animal was going to go. So whether that be the reward well or another sand well. And what we were able to find is that as the animal approaches the sort of 2 seconds before it leaves the start box, we can predict with pretty high accuracy that it's going to go to the reward well. And you can see that there's specific cells that start corresponding to that reward well and kind of triggering that prediction. So for this animal specifically, I'll show you its performance graph. Over all of the correct trials, we trained this decoder. And what we can see in the 15 seconds leading up to the animal leaving the start box, in blue, we see that initially we are finding it very difficult to decode where it's going to go. We don't seem to have much information about the reward location. It's around 60%, but as we approach the point where the animal is leaving the start box, we have this increase in performance, and we're able to predict with really high accuracy where the animal is about to go in the box, in the arena. We then compare this to a control, which is where we train the decoder using randomized reward wells and pairing them with neural activity. And we find that obviously we have a dip down to chance level. So the animal is at chance predicting, we're at chance at predicting which one of the 3 wells the animal is going to head to. So what was interesting here is this specific peak at the 2-second period where we're able to correctly decode the reward well of where the animal is going to go. So we looked at this 2-second period across all of the animals. And what we could find is that across all animals, we had largely a high performance, with maybe a few of the trials dipping down, but the majority are very high. And again, we did that same control of shuffling the reward wells, and we found that they were about chance level. What we did in addition to that was then look at the incorrect trials. So this is a trial where instead of the animal heading out to the rewarded location, it heads to the incorrect sand well where there is no reward. And we wanted to see if in those incorrect trials, the animal was still representing the rewarded location, or if it was doing something else. And as you can see from our decoding performance, the animal, we were not able to decode the rewarded location from the animal. Instead, we had basically a drop down to chance level, kind of indicating that the animal is not thinking about the rewarded location in the incorrect trials. We then looked at incorrect trials, but labeled the training data with the sand wells that it actually went to. And again, we find that there is still around a chance level decoding performance of where the animal is going to go. And this kind of implies that there's more of a suppression of activity when you have an incorrect trial rather than the animal simply encoding the incorrect well. So this kind of helps add to our population analysis. And the next logical question is, well, if we can decode using that 2-second period for the correct trials using start box activity alone, what's going on in the start box? So that's the next thing that we did, was basically take all of our neurons firing throughout a trial and order them by their peak activity, their maximal activity. So here you can see in the start box activity, the neurons that are firing leading up to the animal leaving the start box. And then you can see the neurons that are firing in the arena, and it's all in one sequence. The first question we asked ourselves was, are the cells that are firing in the start box also firing in the arena? Therefore, just simply replaying or pre-playing where the animal's about to go. In some trials, we found the answer is yes. You know, in some trials, we had around 50% of the cells firing the start box and the arena. But in other trials, we found no. In this trial, for example, we have cells that are firing in the start box, but aren't firing in the arena. And so the next kind of question we got is, well, if we've got cells that are firing the start box that are actually telling us where the animal is going to go, but they're not firing in the arena in the trial, what do they represent? So this kind of leads into our future directions and what we're sort of moving towards with the paper that we're aiming to put out for this is understanding these cells coming from these neurons firing within the start box. What does the activity represent? And we've got some ideas that are sort of floating around, and we have a good understanding of where to sort of proceed. The next question, as Francesco alluded to earlier, is rats use multiple strategies to solve spatial tasks. There's no one strategy of doing things, but two well-known strategies are egocentric and allocentric strategies. An egocentric is one where the animal is navigating with respect to itself, and an allocentric is where it's navigating relative to the environment around it. So we want to start actually seeing if we can separate these two strategies out, and to start asking the question, how is spatial information modulated by task factors, things like the strategy employed to solve a task? With that, I'd just like to say a big thank you to the whole of the group that we've got. So obviously, Richard, myself, and Francesco, we've spoken today, but there's also a lot of a massive team behind us. We've got Simon Schultz over at Imperial College that's really been helping us build our analytical pipelines. We've got Nuria and Kapil, which gave a massive amount of background into the social interaction, social cognition experiments, and have done a huge amount of work on it. We then have Dorothy, as Francesco has mentioned, Mira and Stefan, who all helped, first of all, build the event arena, build the analysis, and then really shape what we've been moving towards. Also, it's worth saying a massive thank you to those that have helped us really get the calcium imaging up and running. We've got Mark Schnitzer and his team over at Stanford that helped us build our pre-processing pipeline, helped us with longitudinal registration, and of course the Inscopix team that have been instrumental in helping us get it set up. So with that, a massive thank you for taking the time to listen today, and we open it up to any questions.
Thank you so much, Richard, Francesco, Rufus. This was a fantastic presentation. Gentlemen, thank you so much for today's participation. We hope everyone who has attended this webinar has found this information interesting and insightful. I hope you will all stay tuned for future webinars. Please feel free to reach out to us on our contact page at www.inscopix.com. I hope you all have a wonderful rest of your day. Thank you all for participating today. It's been a pleasure having you on our webinar. I want to thank you all, and I'm going to go ahead and close this webinar today. Thank you all so much. I really appreciate everyone's time. This has been a wonderful day. I really thank you.
Thank you.
Thank you.
Thanks.
Take care, everyone. Bye-bye.
Cheers.